Well, good evening, everyone. I am delighted to welcome you to this evening's Research Connects webinar. My name is Dr. Heather Cook, and I'm the Provincial Coordinator of Knowledge Mobilization at the Alzheimer's Society of BC. Now, as part of my role here at the Society, I help make research more accessible by connecting researchers and experts in the field, like our guest speaker this evening, with those who use research, like those of you in our audience. And so this evening's presentation is the second in a new webinar series called a Research Connects webinar. I'm coming to you this evening from the sunny, albeit smoky Okanagan. And so I would like to acknowledge that the land on which I'm standing this evening is the traditional, ancestral, and unceded territory of the Silks Okanagan Nation. And I also acknowledge that you're joining us this evening from near and far, and want to acknowledge the traditional owners, past and present, of those lands as well. Now, before we begin, please note that you can see the meeting controls by clicking or tapping at the top or the bottom of the meeting window. And because this is one of our more formal presentations, we will be keeping everyone's cameras turned off and the microphones muted. But we definitely want to hear from you. And so I encourage you to use the chat box to share your questions and thoughts as we listen to Dr. Alex Henri-Bhargava. And the chat box can be opened by clicking on that little dialogue bubble that's found on the meeting controls. We'll be monitoring the meeting chat, and we'll put your questions forwards to Dr. Henri-Bhargava once he's finished his presentation. So on that note, it gives me great pleasure this evening to introduce our guest speaker, Dr. Alex Henri-Bhargava. Dr. Henri-Bhargava is a neurologist with fellowship training in behavioral neurology, as well as a master's degree in health practitioner teacher education. He's based in Victoria, where he is medical director of the Neil and Susan Manning Cognitive Health Initiative, which is a clinical research program to integrate leading edge research and care for patients living with cognitive health issues on Vancouver Island. And his research interests include dementia and cognitive disorders, clinical trials, health system improvement, and medical education. Welcome, Dr. Henri-Bhargava, and it's over to you.
Thank you, Heather, for the kind introduction. I'll do one other piece of housekeeping that Dr. Pechora Fuller from University of Toronto, who researches hearing, taught me, is that in nice versions of Zoom, like the Alzheimer's Society has, there's an option at the bottom to show captions. So if anyone is hard of hearing and is having trouble, you can put the captions on to read along as we're going, and through the magic of technology, they will translate my words into text for you. So I will share my presentation here. So thank you all for attending and sharing a weekday evening with me to learn about Alzheimer's disease therapeutics. I'm always impressed by people who have a thirst for knowledge and show up at these things. And of course, we've gotten more used to the webinar format thanks to the pandemic that we've all lived through. I will also acknowledge that the land that I work, play, and live on is within the ancestral territories of the Lekwungen people and on the territory of the Métis Nation of Greater Victoria. This is a First Peoples House at UVic, which is a wonderful space of learning and sharing. And I will disclose that, as Heather mentioned, I conduct clinical trials, and clinical trials are expensive things, and they're often funded by pharmaceutical companies who are trying to develop new therapeutics. I've also received some fundings from non-corporate entities as well. I'm going to be speaking about developments in clinical trials tonight, but I will not highlight any of the trials in which I have personally been involved. And the other thing I would say that mitigates the bias to a certain extent is that the payments for the clinical trials under my direction are provided to the clinical trials unit at Island Health and not to me directly. And it's not a lucrative business to be doing clinical trials by any stretch of the imagination. So really my aim in participating in clinical trials is to try to expand options. But obviously my bias is that I do believe that pharmaceuticals and therapeutics are going to play a role in the treatment of Alzheimer's disease. And there's some people who say that, this is a complex condition. It's not even necessarily maybe one disease, and maybe we won't get therapeutics. And my bias is that we will. So that's the bias I'm bringing to the talk this evening. So as part of that, I'm going to give you an overview of what we understand about the pathology of Alzheimer's disease and what this really is, a general overview of where drug research has come in general in the past 2 decades. And then I'll focus on what I think a lot of people want to hear about is the recent anti-amyloid therapies that have been approved in the United States, and when and how they might come to Canada, and what the pros and cons and really the approach to using these new drugs might look like. And I'll talk about what's going on, further research in the Alzheimer's disease pipeline in terms of what other drugs are being researched in 2023. So on November 25th, 1901, more than 120 years ago, a 51-year-old woman was admitted who had already been experiencing signs of memory loss for several years. And at this point, her situation was quite advanced, and she was resembling what people at the time termed senile dementia, which was thought to be, well, when you get older, you just have problems with your memory. But she was a bit younger than the other patients, and she could remember her name, Auguste, but she couldn't remember her last name and couldn't remember her husband's name. And in fact, she wasn't sure if she really understood the question when she was asked. She could name certain objects, but then when she was asked to write her name, she tried to write Mrs. and then she forgot the instruction, and it was necessary to repeat every word. And so the neurologist examining the patient, Alois Alzheimer, wrote that she was exhibiting an amnestic writing disorder, a problem with her memory when she was writing. Almost 5 years later, the patient passed away, and the persons taking care of her had remembered Dr. Alzheimer's interest in her clinical case, and told Dr. Alzheimer that the patient had passed away, and would he be willing to examine her brain. And this was when methods for looking at the brain under the microscope were just starting to be well developed, and he looked and he saw that there were changes on her brain. After more cases were described, Emil Kraepelin actually named the condition Alzheimer's disease. Alzheimer wasn't proud enough to call it my disease in that sense. And what Dr. Alzheimer saw when he looked under the microscope was that there were two things. There were what he was at the time terming miliary plaques, little plaques of junk that were accumulating outside of the neurons, which are the major brain cells. And so this is what it looks like under the microscope with silver staining. And then with silver staining, he was also noticing that within the neurons, there were these tangles called neurofibrillary tangles. So he made the terms neurofibrillary tangles and amyloid plaques. And for the next 80 years or so, that's pretty much what we knew about Alzheimer's disease. And so it became increasingly recognized that this syndrome, where people have a gradual loss of cognitive function, predominantly starting with memory, was due to a buildup of these intraneuronal fibrillary tangles and these amyloid plaques outside of the neurons. And in the early 80s, scientists discovered that the chief component of plaques was a protein called beta amyloid, and the chief component of the neurofibrillary tangles was a protein called tau. And ever since then, these have been focused on as potential targets of treatment for Alzheimer's disease. But of course, how the amyloid plaques and the tau tangles begin to develop and what their role is in terms of directly causing the disease is still a matter of research to this date. What we do know is that on average, there seems to be a staged buildup of the deposition of both the plaques and the tangles. And so if one looks at the plaques, early on, these plaques are deposited in the more primitive parts of the cortex. And as the disease progresses, they are deposited in more widespread areas of the brain, but relatively sparing areas of the brain important for motor control and sensation, which is why this disease affects cognitive functions much more than it affects physical functions. And on the other hand, the neurofibrillary tangles also have a ordered deposition where they are concentrated initially in the hippocampus and the other structures in the medial temporal cortex. The hippocampus is a structure that's very important for the ability to form memories and to be aware in space. And so that explains some of the symptoms of Alzheimer's that have to do with loss of memory and loss of orientation, because the parts of our brain that are important for that are filled with these tangles. And as the disease progresses, that area of the brain becomes filled with even more tangles, but tangles are seen more widespread throughout the brain. And so we understand now that this is a disease that progresses in terms of the parts of the brain that are affected with these changes, and that it appears that that progression can help to understand the progression of symptoms in typical cases. Of course, there's a good percentage of people who don't have a typical presentation of Alzheimer's disease, and instead of demonstrating symptoms of memory loss, they may experience language changes, or they may experience changes with visual perception with something called posterior cortical atrophy or Benson syndrome, or they may experience changes with behavior in what is termed the frontal variant of Alzheimer's disease. And the reality, when we're looking again at the pathology, at what's happening in the brain, is messy. There is controversy surrounding whether beta amyloid and tau are the actual offenders, or whether they're a byproduct of a whole host of things that are causing the damage. And more importantly, up to 50% of patients who have Alzheimer's disease on autopsy have other degenerative changes in the brain. They have signs of other dementing illnesses, such as dementia with Lewy bodies or frontotemporal lobar degeneration, or a good portion of patients have cerebrovascular disease as part of that. And so there's this concept that maybe one neurodegenerative illness begets another, and so it's not always quite so clear. So we have lots of questions regarding the pathology and the cause of Alzheimer's disease. One thing that we know, if I go back to this slide here, is that along this progression of sequential deposition of amyloid plaques and neurofibrillary tangles, those processes start to happen for years, perhaps even a decade or more, before people actually start to exhibit symptoms. So the brain of most people can compensate for the accumulation of Alzheimer's disease until a point at which there's enough disease present that it overwhelms the brain's capacity to compensate, and patients start to show symptoms. So perhaps one of the ways to stop this disease is to be able to do research in people who are not yet exhibiting symptoms, or who are at the very, very beginning of exhibiting symptoms. We don't know whether these changes really happen in as orderly a fashion in most people as the models suggest. And we don't know if these changes occur in the same way in everyone. And in fact, we suspect that they don't occur in the same way in everyone. And how do we account for mixed pathologies? The other real mystery is that some normal people, especially octogenarians and nonagenarians, if they pass away and have a brain autopsy, have very significant late stage Alzheimer's disease type changes in their brain, and yet they did not exhibit any cognitive symptoms. And there are new players that are being found. There's a protein called TDP43 that Dr. Alzheimer could not see with his staining techniques in the early 20th century, but that with new staining techniques we can find. And what we see is that people who have TDP43 accumulations are much more likely to have had symptoms associated with the plaques and tangles than people who did not have TDP43 accumulations. And of course, the billion-dollar question is, what triggers this disease in the first place? And all of these questions had led some people to say that, you know, Alzheimer's disease may just represent something that happens to the brain, and it's just happening more quickly in some people than others, and it's not really a disease, and we shouldn't treat it as a disease. And I'm showing a picture of Dr. Peter Whitehouse, who was early on a researcher in some early drugs in Alzheimer's, and wrote a quite popular and thought-provoking, if not slightly controversial book, where he argued exactly that we should stop looking for drug treatments because we're trying to basically treat a symptom of aging rather than disease. I would say that even if this is a form of aging, it's very cruel that in some people it happens quite early. I have patients who are in their 50s. I have even patients who are in their 40s who have Alzheimer's disease. Um, Thankfully, this disease does not hit many people in their 40s and 50s, but it does. And it's very devastating in people in their 40s and 50s, just as it's devastating in people in their 60s, 70s, 80s, and 90s. So even if we are dealing with a form of brain aging, we're certainly not dealing with a form of brain aging that hits everyone the same. And we all know people in their 90s or even centenarians who are still doing very well. So I think that the idea that we should just give up doing research is not one that resonates with me at this point. How we diagnose Alzheimer's disease. So I talked to you a lot about these microscopic changes, these plaques and tangles, but in fact, we diagnose Alzheimer's disease most of the time not on pathology. So usually we use a shortcut. When we say that someone has Alzheimer's disease, what we often mean is that they have a dementia syndrome whose symptoms are most likely to match Alzheimer's disease were we able to look at their brain under a microscope. And so we diagnose it based on the pattern of difficulty that's reported by patients and their carers. So do their symptoms sound like the symptoms that people who have Alzheimer's disease have? We look at the pattern of difficulty that they demonstrate on cognitive tests. And here you see examples of the famous clock drawing test that some people are familiar with. And we can see how somebody with early symptoms has some trouble with planning the clock, but it's still reasonably good as a clock. And somebody who has later Alzheimer's has a much more disorganized clock. And we look at clues on brain scans. And so in some persons with Alzheimer's disease, we can see shrinking or atrophy of the brain caused by the accumulation of particularly the neurofibrillary tangles. So here you see examples of people with different degrees of atrophy of the hippocampus, that area that I said that is quite important for memory. Here is the highest grade of atrophy, and you can see a large space here and a shrunken up little raisin where the hippocampus was, compared to a normal patient who has a much plumper hippocampus and only a small space taking up space here. The challenge is that the degree at which someone will have obvious atrophy on their scan by the time they're showing symptoms varies a lot from individual to individual. So we often see patients who have some degree of significant symptoms, and yet they only have very mild atrophy on their scan. But when we see significant atrophy and it's matching with the patterns that on their cognitive tests and on their history, that can give us some clues. Sorry, that's the wrong way. So we've built up this model now where we actually think that there is a buildup of amyloid beta and tau that occurs quite some time before we start to see that atrophy on the scan. And that atrophy on the scan starts to happen quite some time before people exhibit symptoms. On average, not everybody follows the same track. And so the idea is that if we can measure amyloid and tau, we may be able to catch people who are starting to have pathological changes early on in their disease trajectory. And if we can measure amyloid and tau and look at things like MRIs, we may be able to be even more certain about what we're catching. So we do now have the ability to image the presence of amyloid plaques using a technology called a PET scan. The problem is that the type of radioactive tracer that's required for the amyloid PET scan is very difficult to manufacture. It's very expensive. And in Canada, there is no province where an amyloid PET scan, which costs about $5,000 per person, there's no province where this is reimbursed as part of routine medical care. So in Canada, we only have access to amyloid PET scans in clinical research studies. And I'll say for me on the island, we really don't have access to it because by the time the tracer gets to the island, it has degraded and we can't use it. So even in our research trials, we have to send patients away to try to get a PET scan. And we now also have PET scans that can detect the tau neurofibrillary tangles. There are some experimental scans that can detect markers of brain inflammation, which is another aspect of the pathology of Alzheimer's disease that may explain why two people with the same burden of tau tangles and amyloid plaques may have different levels of disease. The amount of inflammation around the tangles and plaque may explain some of the difference, and we can now image some of that inflammation. And in fact, drugs that target inflammation in the brain in a number of ways are now being tested in clinical trials as well. We also have scans that can measure what's known as synaptic density. So that's the connection between the brain cells. And so before we start to see atrophy and shrinking of the brain and degeneration of the brain cells, we might start to see a loss of connections between the brain cells. And so again, this might give us a window into early detection of the disease. And the newer tests are much more in the research realm, whereas some of these tests are starting to be used in clinical practice. We also now have the capacity to measure the amyloid protein that leads to the plaques and the tau protein that leads to the tangles in the cerebrospinal fluid. And we now have access to this for clinical purposes in British Columbia. This is thanks to the tireless work of Dr. Mari DeMarco, who's a clinical chemist at St. Paul's Hospital, who led a study called Impact AD that has published a few papers and will be publishing some new papers soon to demonstrate that being able to make accurate diagnoses in highly selected patients has helped change medical management for them, has been something that has been helpful to those patients, and that has made economic sense from a health economics perspective. The bottleneck, of course, is that this is a special procedure that requires time and experienced hands, and there are not many people who are available to do this procedure, particularly to do it with the protocol required to analyze these proteins safely. And so right now, this is being used in exceptional cases where there's a real diagnostic dilemma, and we're trying to understand, are there plaques and tangles here, or is there something else going on? So an example might be somebody in their mid-50s who is profoundly depressed, and the question is the memory loss a symptom of their profound depression, or is it a symptom of young onset Alzheimer's disease? That would be the case. We wouldn't do this test at this point in somebody who had typical types of symptoms, a brain scan showing hippocampal atrophy, and typical types of test results, but we would maybe consider this test when there is something unusual or atypical about the diagnosis. So how we diagnose Alzheimer's disease in the clinic is shifting away from just looking at the scans and the pattern of difficulties to being able to directly measure what Dr. Alzheimer was able to see on his microscope back in 1906, the plaques and tangles. And so the future will allow us to more accurately diagnose more patients. And in fact, we're hopeful that through the work of Dr. DeMarco and others, we may eventually not need to sample spinal fluid, but we may be able to detect much smaller quantities of these proteins that circulate in the blood. And if we had a blood test, wouldn't that be much easier to access? So I've given you kind of a long introduction, but this will hopefully help you to understand some of the emerging treatments and how they works. So right now, the approved treatments for Alzheimer's disease only treat the symptoms. They do not slow down the disease processes. And they essentially consist of cognitive enhancers, which are cholinesterase inhibitors and memantine. These may improve cognitive function by helping the healthy parts of the brain essentially to function a little bit better. They are the only medications that were developed specifically for and still used in patients who have dementia. And they may have very small effects on important things like whether you can delay placement in residential care, for example. We also use other medications to treat behavioral and psychological symptoms. Most of these we use what is known off-label, meaning the drug was not designed for this purpose, but we know it might be helpful, and so we use it anyway. So an example is the use of an antipsychotic medication that would have been developed to treat schizophrenia, to treat symptoms of psychosis in someone who has advanced Alzheimer's disease. And even to get to our 4 cognitive enhancers here, for each one of these drugs that was approved, as of the mid-2000s, 120 drugs were tested out for each drug approved. And of those 120 drugs, 34 had gone into some phase of clinical trials. And this means that only 0.5% of all the drugs tested to be possibly effective for Alzheimer's disease ended up actually being effective in any way. Whereas the average for the pharmaceutical industry across all conditions is 4%. So usually you have to test 15 drugs for one of them to be a success. In Alzheimer's disease, it was 120 drugs. So this reflects the complexity of the condition and the incompleteness of our understanding of the mechanisms that drive the illness. But if you're a glass half full kind of person, we learned a lot about the disease itself from studying the failed drug trials. We learned about the biology. We learned about biomarkers of the disease. We learned about how drugs may interact in the brain of a person with Alzheimer's disease. This is an example of some of the early studies with the amyloid PET markers and the first anti-amyloid drugs, where we saw that you could clear out the amyloid from a person's brain and yet not stop the Alzheimer's disease. And that's when we realized that perhaps we were treating people too late. If we were treating them by the time they had very significant symptoms, the disease had already set in and maybe we needed to treat people earlier. So we are potentially entering the era of disease-modifying treatments. One thing we have learned about these beta amyloid plaques, these miliary plaques that Dr. Alzheimer saw, they're formed of this protein, as I mentioned, called beta amyloid. Beta amyloid starts from a protein that is inside the membrane of the brain cells called the amyloid precursor protein. And we make tons of this stuff, and we don't really understand exactly what this stuff is doing in its normal biology, which helps to explain why we don't understand what it's doing in its abnormal biology. But what happens in Alzheimer's disease is this amyloid precursor protein gets chopped up into little bits called these monomers. And these monomers accumulate into little clumps called oligomers that then accumulate into long chains called fibrils, and the fibrils build up and develop the amyloid plaques. And the early therapies that were developed were very good at clearing amyloid plaques. But as we've studied the biology, we start to understand that the fibrils and the oligomers are likely more toxic to the brain cells and are causing the disease, whereas the amyloid plaque is like the final junk at the end. So the amyloid plaques is kind of like if you stumble along cold coals and you would say, well, gee, there was destruction here, it must be the coals, but it was actually the fire burning that was the real problem, not the end coals. And so newer therapies are trying to target the flames rather than the coals. The therapies that were developed to try to clear out the amyloid are antibodies. We make antibodies to fight against infection. And for many years now, we know how to make synthetic antibodies in a lab, and we can direct the thing that the antibody recognizes very specifically. So instead of an antibody recognizing a specific virus or a bacterium, we make antibodies in the lab that can recognize proteins. And in this case, we make antibodies that recognize beta amyloid proteins. And then that creates an immune reaction, and the immune system comes and chops up the amyloid, essentially. So instead of directing antibodies that recognize the amyloid plaques, more recent anti-amyloid therapies have looked at antibodies that target the fibrils and oligomers. And one such drug is this drug aducanumab. This was the first anti-amyloid agent to actually be successfully marketed. So all of the other such antibodies had failed to demonstrate efficacy, in part because they were maybe targeting the full form plaques, and in part because those studies were probably done in patients who were too late into their disease. Some of the studies were done before we had the capacity to measure amyloid in the spinal fluid, for example. And so some of the patients may have been misdiagnosed and had no amyloid in their brains to begin with. But aducanumab is the first treatment where they claim, and we'll talk about this in a bit more detail, that there is efficacy in not only just clearing the amyloid, but making a change. What is unfortunate is that all of our anti-amyloid drugs, to a certain extent, cause a complication called ARIA, which is amyloid-related imaging abnormalities. So basically changes that we see on an MRI scan uh, in response to an immune reaction against the amyloid. And that can range from brain swelling. And so I'm sure many of you are not familiar with looking at an MRI scan, but all this white here are areas of the brain that are swollen. And you can see the normal half of the brain here. There's space between the folds, and here the brain is all swollen and there's not much space. And then bleeding, these little black dots represent areas of microscopic bleeding that are seen. And sometimes these ARIA are asymptomatic. They're just something we find incidentally on the MRI. Sometimes they cause mild and transient symptoms that may resemble a warning stroke or a transient ischemic attack, but sometimes they can cause very significant symptoms such as seizures, stroke-like symptoms, and in some cases even death. And so this is a real significant side effect that we need to be worried about. In aducanumab, there were two phase 3 trials. Phase 3 is a phase of clinical trials where we're trying to demonstrate whether a drug actually works. What was interesting was they were very cautious trials initially where the dose was quite low to try to reduce the risk of these ARIA changes. And midway through the study, the protocols were changed to allow a greater dose because they weren't seeing quite as much of the ARIA problems as they thought they would. So the trials were actually stopped early because they said, well, we're not seeing the clinical effect we want. Although biologically, aducanumab was able to reduce the amyloid in the brain as measured by spinal fluid or PET scans, but this didn't seem to translate to a clinical effect. The manufacturer did something that is a bit unusual in the world of clinical trials, where instead of saying, well, this didn't work, we're going to go back to the drawing board, they did some reanalysis of the trials that they had claimed showed it didn't work, and they only analyzed the patients who were on the high dose. And then they said, well, when we looked at that, it seemed that at least in one of the trials there was a response. But this is a bit of an unusual thing to do, as I say, because it's not following the pre-planned analysis of the clinical trial. Despite that, they submitted to the Food and Drug Administration in the States, and the FDA convened an independent advisory panel. And here's where, again, something unusual happened. The independent advisory panel recommended against approving the drug because they said, we don't have enough evidence that this really works yet. We need another study. And yet, the FDA ignored their own advisory panel and they approved aducanumab for treatment of Alzheimer's disease under the accelerated approval pathway, which is a pathway for approving drugs for which there are no current treatments. And this was enthusiastically endorsed by the Alzheimer's Association. And I can understand the perspective of Alzheimer's patients and the Alzheimer's Association for saying, we just are so desperate for anything that might work a little bit. But at the same time, when there's a lot of questions about sort of not a typical pathway for a drug being approved, that's going to leave lingering questions. And so unfortunately, aducanumab has been tainted by lingering questions about the approval. The FDA approval is conditional on a third confirmatory trial that has to occur within 9 years. And to their credit, the manufacturer has said, we're going to get this done within 4 or 5 years tops. And so maybe in 4 or 5 years, we'll have more or less certainty about the efficacy of aducanumab. It's a very pricey drug. I didn't mention this, but it's an intravenous injection, so it's not an easy drug to take. And for all of these reasons, it was rejected by the European Medicines Agency, and the manufacturer then withdrew the submission to Health Canada. So with the current information available, we are not going to have aducanumab available in Canada. In part, this was because a number of organizations representing dementia experts urged caution because of the lack of efficacy, because of the risks of inflammation and hemorrhage that I've highlighted, because this is an expensive drug, and so it could divert funds from more useful programs, especially if we don't know whether it really works, and it may delay the development of more effective therapies. Well, on the back of aducanumab this year, a study was published looking at another anti-amyloid drug called lecanemab. And unlike the studies for aducanumab, lecanemab submitted to the Food and Drug Agency based on the pre-planned analysis of their clinical trials. So there wasn't some of these reanalyses and things that have cast a little bit of doubt on aducanumab. What they showed is that on the bottom left, you'll see lecanemab was very good at removing amyloid plaques on PET scans. So the placebo group in blue had stable levels of their amyloid plaques, and the lecanemab groups had a marked reduction in the amyloid buildup. How did this translate in clinical practice? Well, this is looking at a scale called the Clinical Dementia Rating Scale Sum of Boxes. And basically, what they showed is a 0.5 difference between lecanemab and placebo. What this means is the Clinical Dementia Rating Scale looks at 6 different functional or cognitive abilities and rates them on a scale of 0 to 3. 0 being no impairment, 0.5 being mild impairment without functional consequence, 1 being mild, 2 moderate, and 3 severe. So 0.5 difference means that in one of those 6 categories, you went from, let's say, mild to mild with no functional effects. And so there's a lot of debate about how meaningful this difference is, even though it is statistically significant. There have also been concerns raised about lecanemab. This was a case report that was published shortly after this paper showing lecanemab's efficacy in a patient who was taking lecanemab as part of an extension study to the original trial, who was treated with blood thinners for a stroke and had catastrophic bleeding. All of these black areas that you see on the MRI are areas of bleeding. All of the white that you see here is areas of brain swelling, and this patient perished because of this complication. And so there are concerns that this may not be a perfectly safe drug. And in fact, we know that it does cause ARIA abnormalities even in patients who don't have these catastrophic changes. There's also the cost. This medication will be probably as expensive as aducanumab, and whether that's worth it really depends on who's asking the question. So in this paper on the bottom right, published in June of last year, researchers at McGill University who were also affiliated with the manufacturer of the drug suggested that there could be an $8,000 to $9,000 per person net savings to the American healthcare system from the use of lecanemab. Another group of researchers who were not affiliated with the manufacturer estimated that no, it would actually be a $2 to $5 billion total drain on the US Medicare program. And so I don't pretend to be a health economist, and I know that health economics analyses are extremely complicated, but certainly the jury is out here where we have 2 different groups of healthcare researchers who come to very different conclusions about the potential costs of this drug. Nevertheless, the application for approval in Canada is underway. The manufacturers have applied to Health Canada as of very recently, I think just last week. A third drug that will likely be approved by the US FDA, but this drug has not yet been approved even in the US, and it is not approved in Canada, is a drug called donanemab, which is another anti-amyloid antibody. The company released a press release last week about the results of this medication, but the full study is going to be presented at the Alzheimer's Disease International Conference in July. So far, this has only been reported in a press release. The study is a phase 3 study of 1,700 participants. What the press release says is that there was a less decline in cognition and function in the group treated with donanemab compared to placebo. And then instead of looking at the average change on that clinical dementia rating scale, what they said is, what percentage of patients didn't decline at all? Because over an 18 months of a study, we know that the rates of decline of Alzheimer's disease vary tremendously from individual to individual. And there are some patients, even without any treatments, who we would expect over 18 months may not have a significant decline on that scale. And in fact, 1/3, 29% of patients on placebo don't show any decline, but that number is quite a lot higher. 47% of patients on the drug did not show any decline. And so again, this might be a clinically meaningful change, but it really depends on how you analyze these data. And so we need to see the actual data, and we need to wait for people to do studies of the study, so to speak. What was really interesting is 52% of patients in the treatment group they did the study for 18 months, but they only took the drug for 12 months because by 12 months, all of the amyloid plaque had cleared as measured on PET scans. And in fact, in another study where they didn't look at the efficacy of donanemab clinically, but they looked at whether it was good at clearing out amyloid, and they compared it head to head against aducanumab, they showed that donanemab was more effective at clearing out amyloid than aducanumab was. So we know these therapies can clear amyloid from the brain, but the real lingering questions is how much does this really matter to the progression of the disease in the long run? And when exactly should we be intervening? As early as possible? Are these drugs safe? And we have real questions lingering about what the exact safety of the drugs are. And more importantly, can we make them easier to administer? Lecanemab, for example, is also an intravenous injection. So is donanemab. Lecanemab has to be given intravenously every 2 weeks. So we're talking about a medication that has the same level of complexity as chemotherapy for cancer. And so because of these lingering questions, scientists and neurologists have at least said, well, if these drugs are going to be approved, we have to think very carefully about who are the appropriate patients that should be offered these drugs. And again, I need to stress that none of these drugs have been approved in Canada yet. But in the States where 2 of the drugs have been approved, what they looked at is they said, well, let's look at the patients who were enrolled in the clinical trials. And in the real world, we should be selecting patients who look like the patients who were in the clinical trials, because at least we know that they're comparable. The challenge is that for clinical trials, in order for us to eliminate confounding factors that can muddy up the results, we often are very selective about the types of patients who come into clinical trials. They can't have heart disease. They can't have cancer. They can't be on this drug. They can't be drinking more than 1 drink a day, et cetera, et cetera. There's lots of criteria in clinical trials. And this allows for the patients that are being studied to be a little bit more statistically clean. But it means that it can sometimes be hard to translate the results of a clinical trial into the real world. For drugs where it looks like, well, there's really, these are easy to take, there's no risks with them, then maybe that's not such a big deal. If a clinical trial in a very specific group of patients showed that vitamin C was good, well, we would say vitamin C is pretty harmless. We might as well give it to someone, even if they don't look like the person in the trial. But for drugs that have uncertain benefit and have significant risks and logistical challenges, we want to make sure that we're targeting the patients who are as close to the ones that were already studied. And so I'm going to highlight some of the appropriate use criteria. One is that the clinical criteria for mild cognitive impairment due to Alzheimer's disease or mild Alzheimer's disease dementia need to be met. And unfortunately, in the clinic, most days, we are not as specific about our diagnoses. And so we would have to encourage physicians to be very specific about their diagnoses in order to be able to select patients who are appropriate for this drug. In order to be subject to an anti-amyloid treatment, you need to know that the patient actually has amyloid. Even if you give them a clinical diagnosis of Alzheimer's disease, for this treatment, that's not good enough. That means they need to have a PET scan. And as I mentioned, we do not have access to PET scans in Canada for clinical purposes. And I think it is extremely unlikely that we will, unfortunately. Or a lumbar puncture. And while a lumbar puncture is orders of magnitude cheaper than a PET scan, the access is still a challenge because the expertise to do this is not widespread. And so that's going to be a real bottleneck if we want to be able to select the appropriate patients. The other thing that was found for aducanumab is that patients with a specific gene mutation are both at higher risk of the ARIA complications, but also at a higher chance of benefiting from the drug. And so we might need to consider doing gene testing for this particular gene and counseling the patients. We don't have routine testing for this gene, and we do not have nearly a supply of genetic counselors who are well versed in this gene to be able to launch this on any kind of a scale. And so that's going to be another bottleneck if we were to go with a treatment like aducanumab. And then the other thing is that there are very strict criteria on the MRI to look at patients who may be at risk for the ARIA changes. And that requires radiologists who are extremely familiar with these ARIA changes, and most radiologists are not. So yet another bottleneck. When looking at lecanemab, which has now also been approved in the United States, there was also a paper for appropriate use criteria for lecanemab. The list is very similar to aducanumab, but in the paper, they also listed, well, what are the resources needed by a medical center that wants to be able to prescribe lecanemab? And I'm going to highlight areas where we have very little or none of this in British Columbia. So we need clinicians that are skilled in the assessment to be really precise with the diagnosis, and we have a shortage of specialists and trained generalists who are skilled in that assessment. We need people who are expert in the identification of ARIA, and there's not many of those around. We need people who are skilled in lumbar puncture or PET scans, and we have very few of those. We need the resources to do the gene testing. We need genetic counselors. We need people who are experienced with the drugs. And really, I would say the only people who are experienced with this category of drugs in general are those of us who have participated in clinical trials in BC. And I can unfortunately count that number of people on one hand. And we need settings that where infusions can be made every 2 weeks. So as I mentioned earlier, these drugs are really complex. They're on par with chemotherapy. If tomorrow a new very challenging to use chemotherapy drug comes up for a certain cancer, there's going to be lots of logistical concerns, but there will be a system that's already primed to deliver that drug. There's the BC Cancer Agency. We do not have a BC Dementia Agency. We have a system of patchwork clinics in some cities, and in many cities, the people who are seen who have dementia are seen by their general practitioners. And you can imagine that a GP's office doesn't have any of what's on the screen here. So these are not drugs that a GP will be able to just write a prescription for and send somebody on their way. So we really need to get our medical system set up to be able to deliver complex therapies for Alzheimer's disease. And so we are going to be running into an election cycle at some point. I think it's going to be really important to put this on the radar of our governments because right now it's not. And in fact, this is not just my personal opinion, but focus groups, advisory boards, and research studies have demonstrated these issues as being a real problem, a challenge for being able to roll out these new therapies for dementia. And most concerning in this paper, they identify that Canada of the 10 leading industrial countries is the least prepared in terms of infrastructure and person power. So I've gone over time a little bit. I'm just going to talk very briefly about some other developments on the horizon and leave a few minutes for questions. So we're not stopping with these 3 drugs. And in fact, we don't even know whether they're going to come to Canada for all of the reasons that I've mentioned. As of 2022, there are 143 unique drugs in 172 clinical trials that are being studied. 72% of these are targeting pathologies other than the amyloid plaques and the tau tangles. So we're looking at everything else that's going on in Alzheimer's disease. You can see here mechanisms of action, inflammation, genetics, neurogenesis, meaning stem cells to a certain degree, neurotransmitter receptors, oxidative stress, vascular blood vessel changes, synaptic plasticity. So lots of different mechanisms that are being targeted because we're trying to attack this problem from as many angles as we can think of. So I think there is still a lot of hope on the horizon. The future is that hopefully the next generation of anti-amyloid therapies will be safer, easier to administer. that maybe we will have successful anti-tau therapies, that will have therapies targeting other targets, and that maybe eventually we're going to get to personalized combination therapy. So again, to draw on the cancer analogy, sometimes you go for chemotherapy and you have 3 or 4 different chemotherapy drugs that are targeting different aspects of the cancer cells. We can see a future where you might have one drug that's targeting amyloid, one drug that's targeting tau. If you are somebody who has a lot of neuroinflammation, some combination of drugs that's targeting inflammation. That's where we hope the future is going to head to, but we're not there yet. Participating in a clinical research trial as an individual, the motivation should be to give back to science and to accelerate new discoveries. And, one benefit is that you can access closer disease monitoring than you would otherwise. More importantly, as a system, healthcare organizations should be eager to participate in clinical trials in order to develop the expertise to be able to implement new therapies when they arrive, and to be able to know when not to implement them, to understand the safety aspects, etc., to learn more about the disease from failed trials, and to bring attention of the disease to decision makers. Unfortunately, access to trials is still fairly limited in our province because they're hard to set up, and there's not a lot of centers that do them. So thank you for your attention. I apologize for running overboard, but our moderator will moderate a few questions.
Thanks very much, Alex, for that. And thank you for making a really complex topic easier to understand, as well as making something that's often US-centric relevant to our BC audience. Now we'll move on to some questions that we've had while you've been speaking. And the first one talks about how you mentioned lecanemab has been submitted to Health Canada for approval. So what does that look like? Like, how long does something like that take? Could we see it soon or not? Or how complicated is something like that?
Yeah, I don't know all of the ins and outs of the application process, but I think we would be looking at minimum 1 to 2 years. So there's a myth out there that Canada just tags along to what the FDA might or might not do in the States, and for sure they look at what other regulatory agencies do, but Health Canada takes upon itself to do an independent review of the drug approval process, and we don't have an expedited pathway for orphan conditions as they would in the United States. So I think we would be looking at a minimum of a year process of review by Health Canada, if not a bit more.
Okay, great. Thanks for that. And then if someone was interested in participating in a clinical trial, how would they know what's available or what's available in their areas in Victoria and the Lower Mainland? And how would they go about finding out more?
Yeah. So in Victoria, they can contact the clinical trials unit at the Royal Jubilee Hospital, which is where we do some of the Alzheimer's clinical trials. In the Lower Mainland, it would be through the Clinic on Alzheimer and related disorders. They can also go to the website of the C5R. So if you just type in, C5R dementia in Google, you'll find it. That's a consortium of Canadian Centers for Clinical Cognitive Research. So it's a consortium of many of the academic and non-academic centers in Canada who undertake clinical trials, and their sites are all listed there. There are a few private clinical trial sites that are not affiliated with the C5R. There's also a program in BC called Reach BC. I don't know whether there are many dementia-related studies on Reach BC, but you can indicate any conditions that you are interested in finding out information about trials. And when those conditions are listed on Reach BC, an email is sent to you about that. So those would be some of the ways. Now, one thing I would say about that, that's very disappointing to people who want to participate. As I mentioned, unfortunately, and if I had my way, it would be a little bit different, but many of the trials are overly restrictive about, it's like Goldilocks. You can't have too much of this or too much of that. The porridge has to be just right. And so there are many people who I see a protocol for a trial and I say, this patient would be perfect, but the sleeping aid they're on is not the right one and they can't get off it, or they had some disease that excludes them from being on the trial. So unfortunately, there are a lot of people who would like to participate and who cannot because of how restrictive. And that's something that comes up at international meetings about, how can we pressure the people who design the trials and oversee them at a global level to make them less restrictive.
Okay, great, thanks. And one final question, a bit more of a general question, about often in research you see about trying to include a diverse population and underrepresented populations. Do you have any thoughts on how we could do that, how to increase participation?
Yeah, I hope that tools like ReachBC will be one solution to that, but it's a real problem, and it's not just a problem of outreach. I mean, a lot of underrepresented populations, especially if they're coming from lower socioeconomic backgrounds, people have less time on their hands to look into these to a clinical trial, right? If you're worried about making your next paycheck, or you've got to support your kids or something like that, you've got less time on your hands to worry about clinical trials. But there's also some populations that are traditionally, maybe not intentionally left out of clinical trials, where historically there's been abuse of research, and we need to build better bridges. So, some of the poorer, predominantly Black communities in the United States, there's a lot of mistrust of clinical research there because of some horrible things that were done. And same thing when we do research with Indigenous communities, it's not just about, going to a community and putting up flyers and say, here, be in our trial. It's really about developing a relationship with the community so that, you are known in that community before you can start to say, hey, come and try this experimental drug. And a lot of the funders of the trials don't fund the infrastructure that's necessary to go out. It's changing slowly, but it's slow.
Yeah, I can see that, and how in research in general, building relationships is so important, but something like this, like a clinical trial, such as these, the relationship building is a huge aspect of building that trust.
Yeah. And so for me, the trials I have participated in, the predominant patients come from the clinics that I work in and the clinics that my immediate colleagues work in, because often it's it's a trusted relationship there, where they've seen the poster and they say, well, what can you tell me about this study? Whereas it's not just a total stranger that yeah.
Wonderful. Thanks for that, Alex. And again, thank you for presenting on this. It's such a wonderful topic to learn more about. I'll hand over to Heather.
Wonderful. So yes, just echoing Rakesh's comments there. Thank you so much to Dr. Henri-Bhargava for taking time out of his evening to share his knowledge, and also to all of you for joining us this evening. We would love to hear your feedback. So please take the time to complete the post-event survey that will arrive in your email inbox on Friday. We'd certainly love to hear your thoughts about future topic areas for the webinar series. And also want to flag for folks that our next Research Connects webinar will be in late June, right around June 22nd, I believe. And that will be with Dr. Jennifer Bombush from the UBC School of Nursing, who will be talking about dementia and climate events and the impact of climate change. And in the context of the hot weather we've been experiencing over the last few days, what we might be able to do to prepare for that. So again, thank you very much to everyone, and have a wonderful evening.
Thanks. Good night.